Most of the systems that I've had experience with, data collection systems, require the site staff to alter their normal working routine to provide the data we need for the clinical trial. As a result, what happens is they put it off, and therefore the time between subject visit and the time that the data is submitted to the data management group is unacceptably long, which creates a lot of the delay in getting in the data that we need to be able to make decisions on the trial. We actually put a lot of focus on the tools that we have, trying to ease as much as we can the burden on the site staff. For example, we choose the digital pen technology, which we feel it's easier for the sites. It's pen and paper, so it's something that they're intimately familiar with. We've all used those since high school, so we know what it is. You can see the subject, you can fill out a CRF with a pen, you can dock it, type in your password, you're done. The other thing we do with data collection that's a little different than other folks is we collect a lot of metadata on a trial. I mean, a lot of the information that comes on the CRF is critical to the study, but there's a lot of other things happening on the trial that you need to know what's going on. If you know the metadata and all the other things that are going on, you can really start telling a story of where the study is progressing. We had a situation where a sponsor came to us and they had an original database lock that we had set early on in the study, in the original startup phase and planning of the study. They came to us in May, about 6 months before the database lock, and they said, we need to lock the database 6 weeks early. So obviously everybody is scrambling to try to figure out, is this even possible? But because of the data we collect, we had the data in the database because we collected it quickly and we cleaned it as quickly as possible. We also had all this metadata, and using the combination of the two, we were able to determine exactly what was at the sites, what had already been sent in, what had already been cleaned by the clinical staff and considered closed data. So we were able to calculate exactly how many on-site CRA days we needed between the time they asked us to accelerate the lock to the new database lock date. Luckily, what we found out was to meet the new timeline, they needed essentially 13 on-site days for their CRAs every week up to the database lock. When talking to the sponsor, they had 7 CRAs working on the study, so we knew we had the resources available. Then the challenge became using the data we had to plan the CRAs being on-site 13 to 14 days a week for the next however many weeks it was to actually accomplish the new database lock. The good news is we actually locked the database 3 days ahead of the accelerated timeline, so we were able to perform that because we had all this information that we could assess performance of the people doing the work, how much work they had to do, and plan it out so that they were at the site when they needed to be to get the job done.